Hello students, today we are going to discuss the concept of typification from taxonomy of angiosperms and plant ecology. So we are going to discuss today what is meant by type specimen and its types. So according to the principle 2 of botanical nomenclature, the names of different taxonomic groups are based on nomenclature types. That means when a species is described as a new species by the author, it must indicate which is the type specimen on which the new species is described or based. That means whenever a new species is described by the author, a specimen which is collected originally uh, should be referred as a type specimen, and the description should be based on that type specimen. In the case of a species, the type is an individual specimen, which is a type specimen on which new species is based. In case of small herbaceous plants, actually, when it is mounted on a herbarium, that whole herbarium can be considered as a type specimen. But in case when it is not possible to preserve the specimen, a figure or description can be considered as a type. So the type of the genus is a species, and type of family is genus. For example, in Poa pratensis is the species of the genus is the type species for the genus Poa. So there are 5 types which are given in your syllabus: holotype, isotype, paratype, lectotype, and neotype. So holotype is a specimen or element which is used by the author as a nomenclature type. So nomenclature type, as we have discussed, it is a specimen on which a new species is described by the author. The holotype is chosen by the original author from a single gathering. That means a collection which is made by the collector at one time and expressed definitely at the time of original publication. So for publishing a research paper, uh, a holotype must be chosen from the original gathering or original collection by the collector. So holotype can be a single plant, or maybe a part of one or several parts, or of multiple small parts. In case of fungi or algae, it can be a culture which is preserved and should be metabolically inactive state by different preservative methods, for example by lyophilization or by deep freezing, and this can be accepted as a types. So holotype, it is very much essential to designate a holotype for publishing. So holotype is the specimen or element used by the author as a nomenclature type, which is collected from at a single collection, from a single collection, which is and at one time. Then the isotype is nothing but a duplicate of the holotype, which is collected from the same place, same time, and by the same person. So collection number is also same most of the time and can be differentiated as a B, C, etc. So isotype is the duplicate of the holotype. So whenever author collects many specimens at the same time, so he can designate one among them as a holotype and the rest all will be the isotypes. Paratype is a specimen which is cited in a protologue that is neither the holotype nor an isotype nor one of the syntype. So if a new taxon is collected in a different season with flowers and fruits, then the two collections will be given different field numbers. Out of these two collections, the author will select one collection as a holotype and the isotype, while the next gathering collection which is cited in the protologue will form the paratype. So paratype is a specimen which is collected from the another season, so from the same locality. Lectotype is a specimen or any other element selected from the original material to serve as a nomenclature type when no holotype was designated at the time of publication, or it may be missing. So, if the holotype is lost, it is it is destroyed, the lectotype is selected from the isotypes, and when two or more specimens have been designated as types by the author of the species, the lectotype must be chosen among these types. The last one is the neotype. It is a specimen or other element selected to serve as a nomenclature type as long as all the material on which the name of the taxon was based is missing. So a specimen or any other element selected with no holotype, isotype, paratype or syntype exist. Any specimens other than holotype or isotype included in the protologue is a paratype. Thank you.